Alzheimer's, a word that means so much, a word that affects so many, a word that society is determined to stamp out. Alzheimer's disease, or AD, is an irreversible progressive brain disorder that damages and eventually destroys brain cells, leading to memory loss and changes in other brain functions, mainly the hippocampus and cerebral cortex. Currently, 46.8 million people worldwide live with AD, with approximately 415,000 of those sufferers being Australian. This is due to increase to 540,000 by 2025. So what is Alzheimer's disease? How does it affect the brain? And more importantly, how can this detrimental disease be treated? These are the topics that will be discussed in today's presentation. The brain is a healthy, powerful organ, vital for human function and ultimate existence, having many different components that work together to function optimally. One of these areas is the cerebral cortex. It is an extremely convoluted and complicated structure associated with the higher functions of the mind: thought, reasoning, sensation, and emotion, with each hemisphere controlling certain activities. These areas are referred to as the frontal lobe, involved in movement, memory, and speech; temporal lobe, involved in understanding of sounds and words; parietal lobe, involved in touch and pressure; and the occipital lobe, involved in visual images. The hippocampus also plays a crucial role in understanding and processing various forms of information as long-term memory. In a healthy adult brain, there are about 100 billion nerve cells travelling as electrical charges, which are forming the basis of memories, thoughts, and feelings in an individual. When these signals connect to one another at synapses, they trigger tiny bursts of chemicals called neurotransmitters, which are message senders within the body. Alzheimer's disease changes and destroys this healthy and vital organ by dramatically shrinking the size of the brain, in turn nearly affecting all of its functions. As can be seen in the image here, the cortex shrivels up, damaging areas involved in thinking, planning, and memory. This shrinkage greatly affects the hippocampus, the key area in forming new memories. Because of this shrinkage, the ventricles within the brain, which are fluid-filled spaces, are able to grow larger. However, at a microscopic and physiological level, there have been two most significant physical findings in the brain cells affected by Alzheimer's disease. These are neuritic plaques and neurofibrillary tangles, with another factor being the greatly reduced presence of acetylcholine in the cerebral cortex, the neurotransmitter necessary for cognitive function. While some neuritic plaques are commonly found in the brains of elderly people, they appear in excessive numbers in the cerebral cortex of Alzheimer's patients. They form when protein pieces called beta amyloid clump together. Beta amyloid comes from a larger protein found in the fatty membrane of nerve cells, gradually building up to form plaques. However, it is the smaller clumps rather than the big plaques themselves that cause the most damage. The small clumps block cell-to-cell signaling at synapses, as well as activating immune system cells that trigger inflammation and devour disabled cells. Neurofibrillary tangles, or NFTs, on the other hand, destroy a vital cell transport system made of proteins. As can be seen in the microscopic image here, the healthy areas contain a protein called tau, which helps the system become organised and stay in parallel strands, allowing key materials to move efficiently and easily. However, in the AD-affected tissue, the tau has collapsed into twisted strands. This has hindered the strands' ability to stay straight, making them fall apart and disintegrate. Nutrients and other materials are now no longer able to move through the cells, eventually causing them to die. The development of both plaques and tangles occur in more of a predictable pattern in individuals with AD, beginning in areas important for memory and then spreading from there when compared to normal aging. Even though the specific cause of AD is not 100% known, the linkage in communication blocking properties and disrupting important cell processes, causing death of nerve cells, leading to memory failure and personality changes, creates a strong link between both structures and Alzheimer's. Today, Alzheimer's is at the forefront of biomedical research. 90% of what scientists know about the disease has been discovered only within the last 20 years. This is remarkable progress, however, only has occurred due to the increased funding. In 2013, the federal government of Australia committed an additional $200 million for research over 5 years, significantly boosting funding for Australia's dementia research sector to over $60 million per annum. Currently, there is no cure for AD, with researchers also looking at new ways to treat it. Current drugs mask the symptoms but do not treat the underlying disease or delay its progression. A breakthrough Alzheimer's drug would be able to do this effectively, ultimately stopping symptoms from worsening. There are two current drugs that are used to treat Alzheimer's disease: cholinesterase inhibitors, which work by boosting levels of a cell-to-cell communication by providing acetylcholine, the chemical depleted in the brain by Alzheimer's disease, to the brain. Secondly, memantine, which slows the progression of symptoms in moderate to severe Alzheimer's patients by blocking the effect of excess glutamate. Daily exercise and good nutrition are also used as treatments to stay optimal, such as joints and heart. Alzheimer's is a disease that affects so many individuals directly, as well as millions of individuals indirectly through family and friend connections. This disease can tear the closest of families apart. It is therefore vital that a cure is made as quickly as possible, so no more individuals and families need to be affected by such a dreadful disease.